Good morning, everyone. First of all, I would like to thank the organizing committee for the kind invitation. And also, I'm very glad to have the opportunity to talk about flow cytometry for EV analysis. I'm actually an analytical chemist, so we develop instrumentation and methods for bioanalysis. Okay, from the last session, we already know the EVs are highly heterogeneous, and also it's very small size with a limited content of cargo molecules. Besides this, the physiological environment in which EV exists is also very intricate. There are a large abundant presence of lipoproteins and NVEPs, and their significant overlap in size and density make it very challenging to get a pure fraction of EVs. So actually, single particle analysis emerges as the approach to tackle the challenges I just mentioned. And among a variety of analytical techniques, flow cytometry stands out prominently due to its capabilities in high throughput quantitative and multi-parameter analysis of single EVs. So what it can do is that it can distinguish EVs from nanoparticle contaminants even without the physical separation because you are detecting them individually. And also it can reveal the heterogeneity of EVs, enabling the subset identification and the detection of rare populations. Okay, so the TEM and the cryo-TEM data revealed that the majority of EVs are smaller than 200 nanometer in size. And the conventional flow cytometry, only a very few highly sensitive ones can detect EVs with a size range from 300 to 600 nanometer. So that's really representing the tip of the iceberg. But I also want to remind you that, let's say, if EV is larger than 300 nanometer in size, only makes up 10% of the total population with a concentration range of 10 to the 9th particles per mil in plasma or cell culture medium. So there are still 10 to the 7th particles per mil of EVs larger than 300 nanometer in size. So this quantity is sufficient for conventional flow cytometry analysis, and we also even need to be cautious about the swarm detection. But still you are detecting the tip of an iceberg. And also, we need to keep in mind that compared with a 10 micron size cell, an EV measured in 100 nanometer diameter, the surface molecules is 10,000 times fewer, and the molecular content inside is 1 million times fewer ready for staining. So this all require great sensitivities for the flow cytometry analysis. So flow cytometry analysis for EV encountering many challenges, not only because of the limitation of the instrument, but also the lack of rigid methods or the ambiguities in data interpretation. I'm very glad that there are two articles authorized by the EV FCM working group, and they have been published in JEV, provide background information and detailed guidelines for conducting, interpreting, reproducing, and also reporting EV FCM experiments. Today, I will mainly focus on the development of high-sensitivity flow cytometry. So by incorporating light scattering with strategies for single-molecule fluorescence detection, we at Xiaomei University have developed nanoflow cytometry that enables the analysis of individual EVs of as small as 40 nanometer in size, and the fluorescence detection limit is 3 electronfluor molecules. So by using nanoflow cytometry to analyze EVs is akin to you use conventional flow cytometry for single cell analysis. It can provide you multi-parameter and quantitative analysis. So what can be revealed, both the biophysical and the biochemical features can be measured. For example, can measure the size, particle concentration, purity, refractive index, proteins, evDNA, and lipid membrane, drug content, and surface salicylic acid. And today, because of the time limitation, I will only focus on the size and the protein analysis. So for size analysis using flow cytometry, we need to use monodispersed silicon nanoparticle size standard to build a calibration curve to convert the scattered light intensity to the particle size. And because besides the exponential dependence of particle size on the diameter, the scattered light intensity is also influenced by the refractive index of the nanoparticle. And we can make a comparison. At the 532 nanometer excitation, 60 nanometer polystyrene is about 4-fold brighter than the silicon nanoparticles. And silicon nanoparticle is 3.5 times brighter than EV. So we really need to do the calibration curve, do refractive index correction based on the mean-series calculation. And also I would very much like to show you, based on the mean-series calculation, let's say a 40 nanometer polystyrene beads, the scattered light is equivalent to 69 nanometer or 65 nanometer EVs inside if you excite it at a different laser wavelengths. And because the EV has a core shear structure, so its refractive index varies with size. If we take a refractive index of 1.38 instead of the 1.40 we normally use. So you can see the 40 nanometer PS polystyrene actually equivalent to 80 nanometer EVs. So we really need to do the refractive index correction for accurate size measurement. And also, actually you can use the size distribution for the purity assessment. For example, this is a PFP. And here we compared the separation by using ultracentrifugation and other 5 separation kit. Using, you can see for the samples prepared by UC, you get a very broad size distribution with particle range from 40 to 200 nanometer inside. But for other kits, you obtain from kits, they most likely resemble what the original source, the PFP. So this is one simple way. Actually, we have a poster on Friday that we have developed an algorithm. So actually, you can really use the distribution to predict the purity. And also, one distinguished advantage of flow for EV analysis is high throughput immunophenotyping of single EVs. By concurrently measuring the side scattering and antibody fluorescence, you can quantify the precise percentage of particle or EVs expressing protein marker, so the percentage and also the distributions. And these strategies have been used successfully in clinical diagnosis and prognosis. And so the commercialized nanoflow cytometer have been adapted extensively by researchers globally. They use this to do quantification of protein cargo loading into engineered EVs, and also to profiling the tumor marker on EV surface by using aptamer, and for ovarian cancer diagnosis, and also identification of specific markers of stem cells. Here are just some examples, but all these are done just by single fluorescence detection. In most of the cases, we would like to do double protein analysis because it can not only distinguish from, let's say, tumor EVs from EVs originated from some other normal cells or non-vascular contaminants, but also it can reveal the heterogeneity, let's say, EVs from the cells of different status. But consider the small size of EVs. Mostly the median size is around 70 to 100 nanometer. If you do double protein labeling, the statistical hindrance would pose a great challenge. And we can see here, if if we use PRCP CY5 along with phycoerythrin to label the two proteins, when you do double labeling, you can see the percentage of specific protein positive EV, the ratio dropped. So we see but if we replace the phycoerythrin or those larger size PRCP with, let's say, aloxafluoride, we can see if you use two proteins labeling and each antibody, you label them with different aloxafluoride. Because of the small size, we can see their statistical hindrance can be negligible. So both the ratio and the median fluorescence are comparable with single labeling. Here is another example. So we use a laser, the nano FCM equipped with dual lasers. And we can see if we use aloxafluor 488 to label CD81 and use PE to label CD63. Another one we use two aloxafluorides. You can see when you replace aloxafluoride with PE, you can see an increase in the ratio and the median fluorescence. So by using double labeling, you can get the four different subpopulations. You can, actually it's interesting to find out there are more EVs co-express CD9 along with CD81 compared with CD9 with CD63. So of course, double labeling provides more in-depth analysis of the EV populations. So with the awareness of abundant presence of NVEPs, actually we have done some study. If you get a preparation that is less pure, so we use, this is a laboratory-built nanoflow cytometer with two lasers spatially separated. Then we can label them with the nucleic acid dye and the antibody. So without the cross talk of the spectrum, we identified a population that is can be labeled with DNA, and this fraction, the DNA, attached DNA can be degraded by the DNase 1, but this fraction is insensitive to the detergent treatment, and this fraction doesn't express CD63. So we designate this population as the NVEPs. Of course, more studies are needed to characterize what proteins are carried by this fraction. Okay, in terms of the future direction of flow cytometry, I think for the comparison or consistency, it would be recommended to calibrate the fluorescence intensity into the epitope number. And also, if one high throughput analysis and auto sampler, of course, more lasers and fluorescence channels, statistical hindrance is still there, but with more lasers and more fluorescent channels, you get the user more flexibility in dye selection and facilitate the correlation analysis among different parameters. Of course, the sensitivity determines the scope of application. So we would like to have a nanoflow cytometer that can sensitively detect cover the full size range of EVs, maybe 30 nanometer EVs or even exomer or supermer. And in terms of the fluorescence, it would be best to do single fluorescent molecule, not phycoresin, but organic dyes. Okay, that's all, and thank you.